Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker Anthony, scientific editor of the Molecular Therapy family of journals. This episode features a conversation between Dr. Paloma Giangrande, editor-in-chief of Molecular Therapy Nucleic Acids and Chief Technology Officer at 11 Therapy, and Dr. Maxim Berezovski, professor of Bioanalytical Chemistry at the University of Ottawa. They will discuss a recent article published in Molecular Therapy Nucleic Acids by Dr. Berezovski and colleagues titled Discovery of DNA Aptamers Targeting SARS-CoV-2 Nucleocapsid Protein and Protein-Binding Epitopes for Label-Free COVID-19 Diagnostics. Before we begin, are you ready for the ASGCT annual meeting? Now is the time to register. Unlock 2 discounts when you pair ASGCT membership and early bird discounts. Members save $485 by registering before the February 29th early bird deadline. Tackling everything from CRISPR to cancer, ASGCT is the cell and gene therapy conference that matters most. But you must act fast. This unmatched community of scientists and clinicians is heading to Baltimore from May 7th through 11th. Reserve your spot at the year's most anticipated cell and gene therapy event. Register at asgct.org/2024 before February 29th to maximize your savings. The future of medicine awaits.
So it's a pleasure to have Dr. Maxim Berezovski on our podcast today. As you know, we're here to discuss Dr. Berezovski's article, recently published in Molecular Therapy Nucleic Acids, titled Discovery of DNA Aptamers Targeting SARS-CoV-2 Nucleocapsid Protein and Protein-Binding Epitopes for Label-Free COVID-19 Diagnostics. What you may not know or be aware of is that his article has been receiving a lot of interest lately from our readership and the field at large, given its focus directed at streamlining diagnostics for viral vaccine development, including diagnostics for COVID-19. For those of you who don't know, Maxim has been a longtime colleague of mine and a collaborator. And he is also a recognized expert in the field of aptamer technology, automation at large, as well as diagnostics. And so without further ado, Maxim, it's a true pleasure to have you here today. Let's get started. Can you briefly introduce yourself and tell our listeners what aptamers are and what brought you to work with aptamers for diagnostic applications?
Thank you, Paloma, for this nice opportunity to talk about aptamers. It's my passion for many years. I started to develop aptamers in 2002. So when I did PhD at York University in Toronto, again, simple aptamers for recombinant proteins, then I moved to developing aptamers to cells. Immune cells, like dendritic cells or cancer cells. And recently because of this COVID situation, so we obtained a grant from Canadian government to develop diagnostics based on aptamers. It's our expertise. Aptamers are great, especially for research labs, because like 2 things. So easy to develop. You don't need any cells. You can select aptamers on the bench, and any graduate student and some experienced undergraduate student can develop aptamers through iterative process of called CELEX, where you just incubate, wash, amplify DNA or RNA, and eventually you'll get a pool of aptamers binding to your target. And if you introduce positive and negative selection steps, negative means, for example, you wash out, you remove aptamers to unwanted targets, and you can specify your aptamers to a specific like protein versus, again, a bunch of other proteins, or a specific cell in the presence of other cells. So that's why, again, the first one, it's easy to obtain the aptamers. Second, you can synthesize for low cost, so which is important for academic labs, and incorporate these synthetic DNA or RNA molecules to sensors, to which like any engineer will like, because it's so easy to attach. You actually control every step of attaching aptamer to the sensor. So you know if it's you want to attach to the 5' or 3', or in the middle of the aptamer, you can always do that, and you be sure that like the structure of the sensor you're obtaining, and you can attach multiple aptamers, like 1 or 2 aptamers or trimer of aptamers, and increase the specificity and selectivity. Yeah, that's my answer.
Yeah, great. Yeah, thank you for that great overview. And I guess what you're saying is that aptamers, they're kind of analogous to antibodies, but the advantage is that they are easier to sort of develop and also conjugate, chemically modify, and apply them for many different applications. So it really, you can handle them and can be handled by many different.
Exactly. You can control every step in production of aptamers and every step in making a sensor. And plus, aptamers consider chemicals, not biologics. That's why the regulatory approvals are different. So you don't need to test them on the presence of like DNA or other proteins, because they're, again, chemistry-based synthesized. And so the requirements are less harsh than for example, antibodies.
Not considered necessarily, biologic. Well, that's great, and that's a really good point. And so the many different diagnostic tests that have been approved by the FDA for COVID-19, which are not just antibody-based or serological antibody-based tests, but include also PCR-based detection, right? Why develop a new diagnostic test for COVID-19? And again, if you can highlight those advantages of your approach and technology versus similar technologies for detection of COVID-19 specifically.
First, why we see a commercial SARS-CoV-2 test based on antibodies? I think because of the existed infrastructure, like biotech infrastructure of producing sensors or lateral flow devices or any other sensors based on antibodies. So there are many companies developing the antibody-based sensors for many years. And when COVID happened, so they just switched one antibody to another antibody. So they already had all the reagents ready. They just need to validate and proceed. That's why they were first who approved the antibody-based tests. Mm-hmm. So with aptamers, we need to go through the whole process of approval and setting up the kind of commercial workflow of the new tests. That's why we didn't have time. So we managed just to finish the project and publish it.
Why do you think it was important to develop an aptamer-based approach, given that there were already other approaches that had been approved? I mean, you clearly saw advantages of your aptamer approach, potentially more sensitivity, being able to detect a potential infection much earlier due to the sensitivity. Were there other things in mind that made you go down the route of developing an aptamer-based diagnostic?
I would mention two main reasons. First, so we developed aptamers quite quickly. And I also saw many companies, like aptamer-based companies, they developed DNA or RNA aptamers quite quickly to the SARS-CoV-2 like virus. So it's a matter of actually 1 month or 2 months. Validation of the test takes longer. And that's why the future of aptamers, it's in the fast development of, again, aptamers to unknown pathogens.
Mm-hmm.
So if you already have a selection instrument and DNA sequencer and synthesizer, you can make your own aptamers in a matter of, again, 1-2 weeks. So that's why, for myself, I think that aptamer technology is excellent technology for the doom situations when the next unknown pathogen, unknown virus, unknown like bacterial infection comes, so you can fast produce this recognition elements as aptamers.
So essentially, to summarize, like really a strong, robust platform that can allow you to really move really quickly in the event of a next pandemic, right?
Yeah.
And much faster than current technologies.
Absolutely. And the second reason, Paloma, once I know the sequence of aptamers, I can send you, any of my colleague, the sequence by email. And you can, right in a few days, synthesize the same aptamers I have it here in Canada. You can make it in the United States, in Europe, Japan, Asia, China, any country. So if you have a DNA synthesizer.
Yes.
So it's very convenient.
Yeah, that's brilliant. And so because your technology is quite complex and detailed, but also very elegant, can you briefly describe the process that you use to identify these DNA aptamers, and specifically the process that you use to identify the aptamers to the nucleocapsid and protein of SARS-CoV-2?
The process it was very straightforward. So we took a synthetic DNA library containing like roughly 10 to the power 15 different molecules. Means that each molecule has unique sequence. Unique sequence gives you unique shape. So imagine we had a pool of 10 to the power 15 differently shaped molecules, and we incubated with the recombinant and protein of the known sequence, which we express by ourselves, or you can buy it in any company. So then incubated, washed out unbounders, and keep the bounders, like the DNA molecule which bound to the end protein. Then heat it up or add urea, so we denature the complex, collected the binders, and then we amplify by PCR, and we improve our PCR. We use the called emulsion asymmetric PCR. So it's asymmetric PCR producing single-stranded DNA, but in emulsion to increase the kind of sensitivity and the yield of our product. And we repeated this 6 times, so 6 rounds of selections, and then we obtain a pool of aptamer binding very well with the nanomolar affinity around I think 5 or 7 nanomole apparent KD to the end protein. Sequence the entire pool by next-gen sequencing and pick the most representative, means that the sequences with the highest abundance. And we pick 5 clones and test them how well they bind to the end protein and other mutants by technique called BLI, so biolayer interferometry. So it's very straightforward technique where you can see the binding curve over the time, so you can measure the KD and also rate constants like Q on and Q off.
And those KDs that you just mentioned, those are pretty very tight binding of those sequences that you identify. Yeah. Can you say a little bit more about why you chose to focus on the end protein as your target for your aptamer, as opposed to some of the other tests that had been approved that went after the spike protein of COVID-19?
To be honest, we started 2 selections in parallel, to end protein and to the spike protein. And actually, spike protein we did to RBD and to S1 protein as well. And protein we obtained the aptamer faster. And then we also realized that in case of the sensor, so the N protein is not the bad protein because, first of all, it's very conservative between the different mutants of the SARS-CoV-2. Second, it's actually roughly 10 times more present in the virus than spike protein. So around 1,000 copies of N protein per virus particle, and around 72 copies of spike protein per virus particle. So roughly, again, the ratio is 1 to 10. So that's why for diagnostics, actually, when you have same number of virus particles, you will have more N protein. That's why your sensor will be better and will be more sensitive to the number of virus particles. So that's why, again, we finished the project faster, and we also, now we are in the process of submitting an article for aptamers to spike protein as well.
That's great. But again, the N protein, just to wrap up, highly conserved, less likely to mutate or alter,
yes.
Which if the virus were to mutate, you don't lose the ability to use your diagnostic test and have to develop another one that's catered for that particular mutant. And then second, it's highly expressed, so obviously your test is more sensitive. So maybe you can pick up even early infections and prevent the spread of the virus, correct?
That's true. Or like diluted, like you can analyze diluted samples.
Diluted samples. So
like we used, for example, we tested saliva. So, and we diluted saliva that to decrease the interference from other proteins.
Got it. So you did test human saliva samples that were infected with. Okay. So do you see yourself taking this this diagnostic into the clinic? Are you seeing yourself partnering with a pharmaceutical company or going into a different direction, maybe on a different? Now that I think the market is quite crowded for this particular SARS-CoV-2. Especially when it comes to diagnostics, do you see yourself going into a different direction?
I would love to commercialize this test, and of course, I'm happy to collaborate with any company. But we did the follow-up study. So once we developed and had the aptamers to N protein, so we actually created a very interesting test. Like we used 2 clones of this aptamers to N protein to different epitopes and use the proximity ligation to ligate the 2 aptamers when they bound to the N protein and produce again a longer DNA product that we can amplify it by PCR. And we develop a test kit when we detect the N protein and viral RNA. Because viral RNA, we can do a reverse transcription and produce also cDNA. And in one vial, at the same vial, we had a DNA from aptamers, like double-length aptamer, and also cDNA from the viral RNA. And we use the real-time PCR to make, again, ultra-sensitive diagnostics of the virus, but based on the protein presence and the RNA presence. So like 2 tests in one vial. So that's our, again, follow-up.
But also lower false positives, right, is what this would give you. Yeah.
Yes.
It's
like a sandwich approach. Hopefully, yes.
Great. Yeah. And so I guess what you also started alluding to, I think, briefly, is that essentially what you're really dealing with here is a platform technology that can be applied not only to develop reagents for detection of SARS-CoV-2, but also potentially against many other types of viruses or other pandemics out there waiting to sort of, happen. So do you see really this platform technology being employed to rapidly develop diagnostics for potential future pandemics? And how quickly do you think that it could move into the clinic? Like, is this something that could happen tomorrow, in a month, a year, longer, or?
Before our conversation, I just Google aptamers in clinics, and I found like 2 FDA-approved aptamers, one in 2004 against macular degeneration disease, and actually the second one was approved RNA aptamer approved last year. So that's why the process is slowly again developing in again increasing number of approved aptamers. For diagnostics, there are again many companies producing diagnostic kits. Many of them actually to small molecules, so toxins or heavy metals. So because aptamers can be developed not just to large like proteins or like viruses, they also can be produced to small molecules or antibiotics. And many companies like now like developing like very like interesting diagnostic instruments and kits based on aptamers, for example, for antibiotics, for toxins in clinical samples or environmental samples. Yeah, it's coming. So again, in academia, almost any diagnostic lab try to use aptamers. So instead of again antibodies. Yeah. So because it's a logical step, when you cannot find a good antibody, the next logical step to look around of aptamer field and like see if there is a existed aptamer, or you can develop quickly your own aptamer.
Yeah, and I guess that's really interesting, because one of the main questions that I think many have in the field is that, as you know, aptamers were discovered in the early 1990s, and the biggest question is, why are there no aptamer therapies out there, based therapies or even aptamer-based diagnostics that are really approved in the clinic? But in your mind, what you're seeing is that we are overcoming hurdles and sort of changing that mindset that we had. We've moved antibodies into the clinic. We've been very comfortable with the idea of using antibodies as drugs, as diagnostics, but we are slowly coming up to speed on aptamer technology. Is that correct?
That's true. And I think the biggest problem with like using aptamers in clinics and diagnostic tests, that we're trying to replace antibodies. And many companies, they asking why we need to replace working antibodies. So my own approach, we trying to find areas where you cannot use antibodies, but aptamers, they do it very well instead of antibodies. And they're kind of unique for these research areas or clinical questions. Like cell select, when you develop aptamers to the whole viruses or whole cells without knowing a priori before they're doing experiments, the biomarkers of cells. And when you develop aptamers, you can select aptamers specific to only one type of cancer versus another type of cancer, or to one type of immune cells versus another type of immune cells. You don't need to know the biomarkers upfront. So when you do the selection, your aptamers will find the molecular difference for you. And then you can use them immediately for the detection, or when you pull down your the biomarkers and use the mass spec to identify the names of the biomarkers, you can actually confirm the binding between those biomarkers and aptamers.
Yeah, that's great. And actually, it reminds me, last time you and I talked, that you had brought up a really good point about the fact that now an approved therapeutic, like an mRNA therapeutic vaccine for COVID-19, mRNA technology and mRNA as a drug had been discovered and described in the early 1960s. And it was not until really 50 to 60 years later that we now have this mRNA-based therapeutic. And so it's possible that aptamers just need a little bit more time to blossom and develop. But you're feeling very confident that we're getting there, right?
Absolutely. And when
on a positive note.
when I recruit students to my lab, I tell them an example for if you're planning to have an expedition to Mars or to the moon, you will not take the entire drugstore with you. So like all thousands of possible like drugs and medications, it will be like too much weight for your rocket. So what do you need? You need to take a DNA synthesizer having 4 bottles of nucleotides, and you can create any aptamer to any target. So it's kind of a universal approach when you synthesize your drug on site. So that's maybe, again, when the aptamers will like applied widely to our life.
I love that analogy. And so I think we can conclude, and especially on that really positive note. And so I want to thank you for joining us today. And I would encourage all the listeners to keep an eye out for further developments and application of your technology. So thank you again for listening, and thank you, Maxim, for joining us.
Thank you, Paloma, and thank you, everyone. And again, I'm happy to develop the aptamer technology and see the one day glory of aptamers in our life.